Alright, welcome back. Welcome back, everybody. Today we have lecture 2. Lecture 2 in developmental biology and stem cells. And today we're going to look at a couple of applications, implementations of stem cells in their ability to produce organoids. And then we're going to look at a little bit of the history of stem cells, when they came about, the initial research, looking at the egg of mammalian cells, and a little bit of history. So, I'm gonna look at my notes because that's important. So, we're gonna look at research that was done in 2016 into the pancreas and the stomach of mice, right? So, regenerative research, right? Now, we said in our definition last lecture that the purpose of regenerative medicine is to repair, replace, or the other one, I forget, of tissues in the body for therapeutic purposes, right? So, in this case, in this experiment, we have a mouse. Mouse with ears. It's a fat mouse today. Right? And they were looking for a cure for diabetes. And in diabetes, for those of us who don't know, you have a dysfunctional pancreas, which is bad at producing insulin, or you're non-responsive to insulin, right? So what they did in this mouse was they took the stomach of the mouse, little stomach, and they reprogrammed. Keyword, reprogrammed. That's an R. Reprogrammed. They reprogrammed some of the cells in the stomach of the mouse to be able to produce insulin. How did they do that? I'm going to need a new pen. So, let's talk about this. So you have your little stomach, right? Stomach. Alright. And you have these little layery things in the wall of the stomach. And at the bottom, new colour. At the bottom of those little fibrils, you have stem cells. Your stomach stem cells, right down here. And they give rise to the stomach lining cells. They're very highly proliferative because your stomach's turning over all the time. Stomach has entirely replaced its surface in about 30 days, right? So what those stem cells are doing is they're generating the cells that live above, up here, right? And they interact with the stomach environment, they process food and whatever. Now what they've done in this experiment is they've taken some of these cells, you extract a part of the stomach from the mouse, and you apply some transcription factors. Now what's a transcription factor? We've never heard that term before. What's a transcription factor? A transcription factor, transcription factor, right? What's a transcription factor? A transcription factor is something that turns on or off a gene in a mouse or a human or whatever. Now that can be a growth factor, signaling protein, something that binds to the genome and turns on or off transcription. Or it could perhaps be an enhancer. Those are also transcription factors. So what a transcription factor does is it causes or inhibits the expression of a gene. That's what a transcription factor is, right? And of course, every cell in your body has the same genome. And so the only difference between those cells is what genes they're expressing. Of course, there are other differences in terms of like what they're exposed to in their little environments and their methylation of the DNA, but we'll talk about that later. The real difference is what parts of their genome they're expressing. And what causes those differences in genome expression is transcription factors. Now, in a particular gene, say muscle cells, in a particular cell, you're going to have factors that the cell itself expresses, which lead to the expression of genes. So they create their own transcription factors. But if we induce stem cell or transcription factors, we can get the cell to behave in the way that we want it to. So, in this case, we've taken stem cells from the stomach, and the stomach and the pancreas are formed of the same tissue layer, and they're very similar. They're kind of located in the same place, and they're both endothelial cells, endothelial tissues, right? So they come from the same layer, and they're very close to each other, and they're quite similar, right? Those stem cells in the stomach and the stem cells in the pancreas, quite similar. So what these researchers did was they were like, whoa, what if we took those stem cells in the stomach and we made them behave like stem cells in the pancreas, right? So how do we do that? How do we reprogram? And this is direct reprogramming. There's also indirect reprogramming, but we'll do this first. How do we reprogram them? Well, we give them transcription factors. So now, we've got our little piece of stomach, we took it out, we exposed it to transcription factors, right? And these little cells now, right, have turned into pancreatic stem cells. Why is that a big deal? Well, they took that little sample of pancreatic stem cells that we generated using the stomach tissue, and we've created what's called an organoid. Organoid. The important word, right? An organoid is a little functional organ that's not got all the structure of a normal organ, but some of the structure and the functional tissues. So what we've done here is we've replaced these and we've created insulin positive cells, right? So up here, instead of producing stomach cells, we now have pancreas cells. Right? We now have pancreas cells. And so, a little sample of stomach, that was once stomach, is now behaving like a pancreas, right? Now, I need a particular word, and I've forgotten what it is, so give me a second. Where did I put my notes? They're in my pocket. Right, so, what did we do? Hypoglycemia, transfection, stomach endothelium, scaffolding, mouse. I'm back. Okay, so we take that organoid and we put it in the greater omentum. This is just a little bit of background knowledge. The greater omentum is a fatty tissue that surrounds the stomach, and it's got a lot of veins, so if we put this organoid into that tissue, it will produce veins going into that tissue and populate it nicely so that it behaves and survives, right? And so, now we have a nice biological assay, physiological assay, right? So if the mouse is able to produce insulin, and if the mouse survives, because we've killed all the insulin-producing cells in the new mouse, we have successfully reprogrammed stomach cells to behave like pancreas cells. Now this is a big deal, right? This is a big deal because this is like the first time that we were able to reprogram an organ to act like another organ. Big deal, big deal. And that is kind of an example of one of the really cool applications of stem cell therapy to like that's where we're at in terms of the scene. Right, let's look at another. Let's look at another application. So let's look at another application. So, you have a brain, probably. What happens if you develop a disease on your brain and there are two drugs that could help your disorder, depending on the diagnosis that you have? And the diagnosis is a little bit imprecise. We don't quite know what's going on. We have a pretty good idea. We've got it down to two things. Now, the two drugs are contradictory and they won't necessarily, you can't put them both in. So you have to make a choice. So either you get better or you get worse, and you kind of got to choose between these two drugs and you don't know which drug is the right drug. What do? What if we could grow a brain? What if we could grow a brain of yours, a little pretend brain, and then experiment with those drugs on your little pretend brain? See how it responds, see if it cures your disease. That's personalized medicine. That's personalized medicine. Right? And so what we've done is you can take a sample of, say, a fibroblast from your skin. We induce pluripotency to create pluripotent stem cells. Induced pluripotent. We can induce pluripotent stem cells, and then we differentiate those into brain cells, and then we create an organoid out of those brain cells. Right? Now we're going to go through a little bit what an induced pluripotent stem cell is and how that's made, and just get a little bit more of an understanding of how that works. Now, the process that we just went through was direct differentiation. We took cells from a stomach and we directly differentiated them using a couple of transcription factors, I think there were 3, to directly behave like a pancreas. That's direct differentiation. In this case, we're looking at de-differentiating. So we take a skin cell, we get it to behave like a pluripotent stem cell. Remember, pluripotency is fully capable of turning into any tissue. The most stem cell-like stem cells, if you will. And so we take our fibroblast, we de-differentiate it, and then we re-differentiate it into a neuron or a brain cell, right? So, let's look at that process. So you have your fibroblast. It's a myofiber. Myofiber, it's got all of its stem cells. Right. So I've taken a sample from your skin, and we get a fibroblast out of it, or we get a satellite cell, or whatever. That's kind of a loose representation of a satellite cell. Something like that. Right. And then we induce it with transcription factors, right? So the transcription factors, you don't need to know this yet, but it's going to be MyoD, OCT4, and SOX2 and SOX4, I think. It might not be SOX2. Those are called Yamanaka factors. Yamanaka factors. And Yamanaka was the first experiment to induce pluripotency, and we're going to go through that in some detail a little bit later. But you take your skin cell, you de-differentiate it, you take it up, the chain to an induced pluripotent stem cell, and then you re-differentiate it into a brain cell. Right, so right after this, right, we get out a nice iPSC, induced pluripotent stem cell. And then we do that into a neuron. Right, so we get our nice little neuron with its axon and dendrites, and it's all very exciting, something like that. Bang. And then, you know, like, in order to neurons don't proliferate, so you're going to need to do some neural progenitor cells, so it can proliferate, and then it can differentiate into your neurons. So there's going to be a neuron progenitor, right? And then it'll differentiate. But the point of all this process is that you can take a little bit of skin, de-differentiate it, and re-differentiate it into a little brain upon which we can test drugs. And that's a big deal, because it allows us to experiment with different treatments for you and to see what different drugs are going to be able to cure your disease. Right, so that's the applications. That's the applications. Now we're going to do a little bit of history. So, when did Darwin publish his evolutionary theory? Not that important. It was 1859. But in 1868, there was kind of the first research into eggs of mammalian cells, looking at, you know, well, the theory for this point in time was that you're kind of formed as a complete little tiny human when you're formed, right? So your whole developmental pathway is just like getting bigger, I guess, which is kind of weird to us now, because we know that you have an egg and a sperm, they come together and it divides and it turns into a whole developmental pathway. This here was the first research into the egg and seeing how that developmental pathway goes, right? And so they looked at the egg and they kind of determined that the egg has, it was called a kernel, but it's actually the nucleus. And they stated, and this was before we knew that DNA existed, they stated that they all kind of look the same. All the mammalian eggs look the same. And so there must be some chemical difference between them that causes them to turn into the different organisms. We now know that that difference is DNA and a whole lot of other molecular machinery. But at the time it was kind of a really good piece of research. Right, then at like 1906, I want to say, 1906, we had the first mention of stem cells by some German guy who I forget his name. And then we had to wait a long time, okay, until 1961, 1963, for Till and McCulloch. I think that's how you spell it, I don't know. For Till and McCulloch. And Till and McCulloch did the first iteration of that mouse experiment that I went through last week, right? So that first experiment was, and you can go back and watch it, but if you want a summary, if you take a mouse, you irradiate it and kill its hematopoietic stem cells, and then you implant hematopoietic stem cells from a different mouse, or that you've taken out of the previous mouse before you irradiated it, that stem cell line will give rise to blood. Now this research was actually off the back of radiation research because of World War II. You know, there's a lot of people dying of Hiroshima and Nagasaki and the nuclear attacks. And so this research was really into that. But it gave rise to really the first investigations into stem cells, which is pretty cool. And so that's our second lecture. So just a quick discussion of some examples and indeed a little bit of history. Thanks guys.